These are your video notes for classification, the start of the next unit. So to study the diversity of life, scientists use a classification system to name and group organisms in a logical, evolutionary manner. So taxonomy, not taxidermy, taxonomy is the science of grouping and naming organisms based on lines of evidence that we talked about in the natural selection and evolution unit. So physical similarities, particularly homologies. Remember, homologous structures, homologies. So homologous structures are structures that indicate common ancestry. Those are the physical similarities we want to use. The analogous structures, remember, analogies are due to convergent evolution and do not demonstrate common ancestry. So analogous structures are not good to use. You want to use homologous structures when you're looking at physical similarities. You can also look at the chemical makeup. You can look at the molecular data, so the DNA sequences or the amino acid sequences for proteins. The more similar those are between species, the more recent their common ancestor. You can look at evolutionary relationships using, well, again, common ancestry, but fossils as well. And you can look at developmental similarities using embryology. But again, DNA is really where it's at. This is where most of our current lines of research have brought us to. It's the most accurate way to group organisms. And there's actually quite a lot of statistics involved for those people interested in redesigning our understanding of the relationship between organisms. Because historically, those relationships were based off of looks, right? Physical traits, and those are not always the most accurate. So Linnaeus started this all the way back in the 1700s and gave us this classification system that we still use. Kingdom, phylum, class, family, or order, family, genus, species. A mnemonic is King Philip came over for good spaghetti. Kids prefer candy over fresh green salad, perhaps. The kingdom is the largest grouping of organisms. It has the most in it. And then as you progress down this list, you get to an individual species, which is the most specific. So here's an example. The animal kingdom, Animalia, consists of anything from sea stars to grizzly bears in this example here. Chordates are your phylum. They have a backbone, spinal cord. Chordata is going to eliminate the sea star because that's an invertebrate. Then you get more specific. You get to your mammals, which is a class. That would get rid of the snake, which is a reptile. Then you get into your carnivores, your carnivora, which is an order. That would get rid of your squirrel, for instance. And then your Ursidae, this is where you get into your bears here. That would eliminate this fox, and then so on until you get down to a specific, polar bear, for instance. Each level of this hierarchy is called a taxon, and the plural for that is taxa. So again, just as you progress down this grouping, you get more and more specific. So for instance, a kingdom includes a whole bunch of related phyla, a phylum includes a whole bunch of related classes, and so on and so on and so on. Remember, by the time you get down to the species level, a species is a group of organisms that can interbreed and produce fertile offspring. Remember, interbreeding is just you can mate with other individuals and make babies, whereas inbreeding is among very close relatives, right? And fertile just means that you can have kids, right? Your kids can have kids, can have kids, can have kids, etc. Oh, and here's our mnemonic. All right, binomial nomenclature. So when we name a species, we don't bother listing off its kingdom and phylum and class and all that kind of stuff. We really only care about the genus and the species. So this is the binomial part of this. Bi means two, so genus and species are two names. And then nomen just means name, is Latin for name. So two names, genus and species. You typically either use italics or you underline both of them, but you always capitalize the genus. So Canis familiaris is your dog, Canis lupus is your wolf, Homo sapiens is humans, and Felis domesticus is cat. Right. And that's just a way for us to communicate these across languages and cultures. Right. If you're communicating scientific findings between people who speak different languages, it can be a little tricky. So we use a common language based off Latin, and that's why we use binomial nomenclature. So which two species are most closely related or more closely related out of the 1, 2, 3, 4, 5, 6 that are up here, right? Well, again, these are based off evolutionary relationships. That's the whole point of classifying organisms. So you're looking for the most similarities as far down as you can possibly go. So all of these are animals, so that's not too useful. But the first 1, 2, 3, 4, 5 are chordates, and the fly is an arthropod, right? So from the phylum level down, your arthropod, your fly is going to be completely different. Insecta, Diptera, right? Blah, 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 blah. All of that's going to be different because the phylum is different, right? So if you're ever doing a problem and you see that like the family for the fly is the same as the family for, you know, a gopher, hopefully that's a red flag because that should all be different all the way up to the phylum. So basically you're just comparing and seeing when the differences start. So flies differences start very early on as the phylum. So that's actually the answer to number 2. I kind of skipped around here. The answer to number 2 is the fly is least related to all of the others. But which are the most closely related? You're just looking for the most overlap. Animal, here they are. These two. Right? They have, everything is the same from genus on upward, except the species, right? That's where the difference is. And this should also kind of make sense because we literally artificially selected wolves to get dogs. All right, classification today, again, is usually based off of DNA or amino acid sequencing. And we're oftentimes rearranging our groupings of organisms because early on, before DNA, we just did this based on looks. And DNA often tells a different story. But based on that information, we like to visualize this, so we make things called cladograms. A cladogram is a diagram that shows the evolutionary relationship among organisms or which are most closely related. So here's an example of why looks aren't the best. So if you had a crab, a barnacle, and a limpet, if you just looked at these and you would say, okay, well, obviously these are kind of cone-shaped shells, and the crab is kind of, it has legs, and it just looks different than the other two. The crab must be the one that's least related. So you might draw a cladogram like this, and you might have the barnacle and the limpet, if you kind of go back in time. They would be the ones to have a common ancestor with each other first, right? This is based off of visible similarities, and this is actually this is wrong. We didn't realize this until after we sequenced the genomes for these. What we did, sorry, the sound effects. What we did is we realized that after we sequenced these, the barnacle and the crab actually have the more recent common ancestor with each other because these are crustaceans. The limpet is a gastropod, which is an earlier kind of off branch of the organisms in this cladogram. And again, you wouldn't know this unless you sequenced DNA. So this is what I mean when we're saying we're kind of going back and revising our groupings and classifications because they were originally based off looks, which are not always accurate, and we're redoing it based off DNA evidence, which is kind of the gold standard here for evolutionary relatedness. So here's a cladogram that shows everything from your jawless fish all the way up through your mammals and your great apes. So the individuals are kind of representative of different groups. So your hagfish is an example of a jawless fish, right? The perch is an example of a bony fish, an osteichthyes. Salamander is an example of a amphibian, lizard, reptile, pigeon, bird, obvious, and then mouse and chimp for mammals. So how do you read this? Well, the first thing is, as you kind of go up, you go forward in time. And actually, the other thing is, it's really similar along this axis too. This is also kind of time. So as you kind of progress, for instance, diagonally along this main backbone, you're moving forward in time. So the oldest organism on here is the hagfish. If you trace its common ancestor with everything else on here all the way back, you get a common ancestor right down here, and that's the oldest. But what you can do is you can look and you can put features on here and deduce what kind of features are present in each of these groups. So the hagfish evolved before Jaws did. Jaws came after hagfish. So everything kind of to the right and up of this point has jaws. So if we go forward, we hit a branch point here. There's a speciation event right here. You have fish coming up this way and the rest of the group coming up this way. But fish and everything else has jaws because jaws evolved lower in this cladogram, right? Again, we're visualizing when these evolutionary events happened, right? Lungs evolved after fish had already branched off in their own kind of lineage, right? So fish and perch don't have lungs, but everything else does. Salamanders, lizards, blah, 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 right? Amphibians branched off before claws or nails evolved, right? So lizards, pigeons, mice, chimps have claws or nails. Then lizards and your reptiles broke off. Then you had a speciation event here with your birds. And notice where feathers are, right? Feathers is unique to this bird lineage, the aves. That's a branch of reptile. It's a branch of dinosaur, actually. So only your birds have feathers, right? But they don't have fur or mammary glands because that evolved after your bird lineage already broke off, right? But everything up from this point has fur and mammary glands. So your mice and your chimpanzees and your humans and all of your mammals, right? So let's do a practice. Let's say what features on this cladogram do pigeons have? What do pigeons have? So there's your pigeon. Well, they've got jaws, they've got lungs, they've got claws and nails, and they've got feathers. They do not have fur or mammary glands, right? What features does a salamander have? Well, it has jaws, it has lungs, but then it breaks off before the evolution of claws or nails. So it does not have claws or nails, it does not have fur or glands, mammary glands, and it does not have feathers, right? So that's how you would read these, and that's how you would determine the structures that these had and the evolutionary relatedness of the species. All of these cladograms are put together statistically using DNA data. And there are completely not independent, but there are lines of biology and professions that are specifically devoted to rebuilding and redrawing cladograms based off DNA evidence. So here's a cladogram for just letters for different species. Who is D's closest relative? So here's D. Basically, you would trace back and find the common ancestor, the first intersection between D and another species, which is shared with C. So there's their common ancestor right there. Right. What do the numbers show? The numbers are showing us speciation events. Right. These could be geographical, behavioral, temporal, etc. Different speciation events. Again, as you kind of go up in this diagram, you're going forward in time such that the top up here is present day. If you had a species like this that maybe came off here, and stopped, it's because it went extinct, right? It did not make it to present day. All right, we did talk about this earlier, but again, classification today is based off of homologous structures because those are the structures that demonstrate common ancestry, not analogous structures, right? Those do not show common ancestry. Again, the gold standard here is DNA data. The more data is shared, or DNA data, DNA evidence, the more DNA is shared between two species, the more recent their common ancestor, right? That's how we know that humans and chimpanzees have a recent common ancestor with each other. Out of all the living things on this planet, chimps are our nearest relatives. And as we sequence more and more genomes, and as we learn more and more about the genetics of other organisms, we are constantly trying to redraw the tree of life and the relatedness between species. So groups like reptiles are almost certainly in the future going to be renamed, and we'll talk more about this if you ever took AP Bio, because there are a lot of genetic quirks popping up that are causing us to kind of rename and recategorize species. Again, none of this is possible unless you understand evolution. And ideally, when you have a cladogram, the branches nearest to each other are lower down on this list, right? So if you have a cat here and a leopard, those perhaps are part, well, they're not perhaps. These are of the family Felidae, or Felidae for feline. And then as you branch out, as you go down the tree and you include more and more groups, so as you include like the wolf here, you've expanded from class, from family to order, right? Order Carnivora. As you expand and you include more individuals, so now you've included like a horse, you've expanded to, there we go, the class Mammalia. And then if you wanted to expand again and include another branch, that would include like your reptiles here, then perhaps you'd be looking at a phylum at that point. And you can keep expanding this and going bigger and bigger and bigger. So this hopefully should make sense. As you get more specific in a cladogram, you're kind of going down this list, and as you expand and include more organisms, you're kind of going up the list. All right, a dichotomous key is a cute kind of little if-then way to figure out what you're looking at. And again, scientists make these to help classify organisms, and you basically just go answer yes or no questions. So here's a quick little example. Let's say that you did not know what this organism here was. We know it's a chicken, but let's just say you didn't. You'd start with number 1 and you would just answer the questions and do what it tells you. So, does it have feathers? It has feathers. Yes, it does. Go to number 2. Number 2 says, it has webbed feet. Well, no, chickens don't have webbed feet. So this thing doesn't have webbed feet. No webbed feet means it's a chicken. There it is. What if you didn't know what the lizard was? You're like, I don't know what this thing is here. Let's do it. It has feathers. Go to 2, or no feathers. Go to 3. Okay, well, there's no feathers on it, so let's go to number 3. It has legs, or it has no legs? Well, it has legs. Go to 4. It has a shell or no shell? Well, no shell, so it's a lizard. Boom, right? Obviously, this is a pretty easy, straightforward one, but that's the gist of how to use a dichotomous key. All right. The classifications that we have today, yes, they include 6 kingdoms, but we also now have what are called domains, and they are above kingdoms. They are more broad than kingdoms. So you have 3 domains, 6 kingdoms. The Archaea domain only has 1 kingdom, Archaebacteria. The bacterial domain only has 1 kingdom, the Eubacteria. Eubacteria are the bacteria that you probably associate with getting sick, right? Archaebacteria are bacteria that can withstand very, very extreme conditions. So although they are kind of around us every day, if you went to like a hot spring or something, then you would only find Archaebacteria rather than both Archaea and Eubacteria. But when we think of bacteria, we're usually thinking of the domain bacteria and the kingdom Eubacteria. The third domain, Eukarya, includes your protists, your fungi, your plants, and your animals. Those are your kingdoms, and those are your domains. This is just a diagram showing the evolutionary relatedness between these kingdoms. One interesting thing here that I do like to point out is that fungi and animals are more closely related than are fungi and plants. A lot of people think that fungi and plants are more closely related, or that fungi are more related to plants than they are to us, but they're actually more related to animals than plants. Again, we would not necessarily know that without genomic DNA evidence. The origin of the bacterial kingdoms and when they branched off of the prokaryotic lineages and the eukaryotic ones is the active area of research, right? But the rest of this we have pretty darn well figured out, that bacteria evolved first, then your single-celled eukaryotic protists, then your plants, then your fungi and animalia. I'm going to let you look over this more or less on your own. But I will do some highlights of the 6 kingdoms. So again, Archaebacteria, they are prokaryotes, they are bacteria. But the point of these is that they live in very extreme conditions. Very hot, very cold, very low oxygen, very acidic, right? Stuff like that. They have cell walls, they're unicellular, they are autotrophic, they make their own food. Some do. And then some are heterotrophic. Some actually need to consume others. They actually break down and help decompose and decay plants and fungi and stuff. Eubacteria, again, another bacterial kingdom. Again, they're prokaryotes, they have cell walls, they're unicellular, they can be auto or heterotrophic. But the point about these is that they cannot survive in extremes. So, I mean, all of your common bacteria that you might associate with getting sick, blue-green algae are actually bacteria. So that's your second bacterial kingdom. Your protista, your single-celled eukaryotes, well, most are single-celled, some are multi-celled like seaweed. This is actually not a plant, it's a protist and it's multi-celled, but most are unicelled, right? This is the kingdom that Leeuwenhoek saw when he looked under the microscope and looked at pond water, right? He saw amoebas and paramecium, and those are protists. Some are autotrophic, in which case they'd be green, like Euglena, for instance. Others are heterotrophic, like amoebas, and they have to consume for food. Fungi. Remember, everything on this point, from this point onwards, is a eukaryote. It has a nucleus and membrane-bound organelles. So most are multicellular, but some, like yeast, are unicellular. They also have a cell wall, but it's made of chitin rather than peptidoglycan, which is what bacterial cell walls are made of, or cellulose, which is what plant cell walls are made of. They are heterotrophic. They do not make their own food, right? They are heterotrophs. They are consumers. They are decomposers, right? Mushrooms, yeasts, molds. So fungi. Plantae, again, eukaryotes, they are all multicellular. There are no unicellular plants. Their cell wall is made of cellulose. They are autotrophic, which means they can produce their own sugar, right? Glucose. They are producers, everything from your simple mosses and ferns to your flowering plants, conifers, right? Plantae. And then animalia. Again, all eukaryotes, all multicellular. They do not have a cell wall. Heterotrophic. Anything from SpongeBob, from sponges, all the way up through your mammals, right? Invertebrates, vertebrates, everything else, starfish, blah, 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 right? All animals. Oh, last little slide here. Coral reefs include they're all living animals, right? These are animals. If you look at coral, they're not plants. Sponges, coral is a type of sea anemone. Fish is zooplankton. But the actual coral itself is an animal. Obviously, fish are, zooplankton are, um, we'll talk about plankton. Plankton is just kind of a floating organism, but those are mostly animals as well. Actually, they are animals, but these are systems of animals. So I don't know why I have this slide on here. It's a pretty slide. All right, I hope this helped, and I'll see you in class.